Center of Research in Medical Devices. That's called as the Curran National University of Ireland, Galway, where he involved more into biological studies and the development of polymer-based microreactors for encapsulating various enzymes. His multidisciplinary profile with core research training in material science enabled him to address multifaceted problems through the development of polymeric systems with advanced functional properties that respond to the inherent disease causing changes in the physiological environment. Throughout Dr. Eiter's career, he has engaged in several multidisciplinary disease-focused research projects with biomedical engineers, biologists, and clinicians to tailor polymeric delivery platforms for therapeutic and regenerative purpose. And since 2018, he's an associate professor with a position at the University of the Basque Country, that is in Spain, and he continues to do his research activities in the development of multifunctional polymer capsules. Professionally, he has been very actively contributing to the scientific community since 2013 with 2 book chapters, 2 patents, and more than 21 contributions in international conferences and over 30 research high peer impact factor journals in the field of material science and engineering and biomaterials, such as Progress in Material Science, Advanced Functional Materials, Acta Biomaterialia, Advanced Healthcare Materials, and Nanomedicine, Nanotechnology, Biology, and Medicine, among others. It is a great pleasure to invite you, Dr. Eiter, today. And to our audience, I must mention that I was highly impressed with the institute and the university Polymat, which I was lucky to go there and present our work on 15th of November with Professor Marcelo Calderon, who is also today as a participant for this talk. I request now Dr. Eiter to actually present his talk, and he might take about 55 minutes to 1 hour of a time. I kindly request to mute your mics. Dr. Eiter, please.
Okay, thank you very much for your kind introduction, Jayan Kandare. They told me that there is a little mismatch between my connection and your connection, so I will try to do the transition between slides as slow as possible, okay? So, well, first of all, thank you very much. I have to acknowledge the organizers of this virtual seminar, and in particular to Professor Kandare for giving me the opportunity to talk a little bit about my work today. So today we'll be talking about the therapeutic use of polymer capsules as microreactors. But before going into the detail of this particular topic, let me very briefly introduce myself so you know a little bit more about my background and which are my main research interests. So, well, I did my PhD at the University of the Basque Country here in Spain in the group of Science and Engineering of Polymeric Biomaterials under the supervision of Professor José Ramón Sarasua. The PhD was entitled Fabrication and Characterization of Lactide and Caprolactone-based bioresorbable and bioactive polymer scaffolds for tissue engineering applications. During this time, what I was mainly doing was to synthesize novel bioresorbable polyesters with specific mechanical properties and specific degradation rates. And then we were using these kind of polymers for the fabrication of 2-dimensional and 3-dimensional scaffolds for tissue engineering applications. I realized, however, at the end of my PhD that my background or my knowledge in cell studies was quite limited. So I decided to do a 2-year postdoctoral stage at the Center for Research in Medical Devices in Ireland, under the supervision of Professor Abe Pandit. And there I had the opportunity to work with many kinds of cells and to study the interaction between these kind of cells and the biomaterials that I was developing. So I had the opportunity to gain some experience in cell and molecular biology that I would say they were vital for my development in the field of biomaterials and biomedical engineering. During that period of time, and also thanks to a short-term stage in the University of Basel in Switzerland, I also started working with polymer capsules for therapeutic applications. So Right now, I could say that I'm following more or less those research lines that I started both during my PhD and during my postdoctoral stage. On the one hand, I'm still working on the development of two-dimensional and three-dimensional scaffolds for the regeneration of tissues. And I'm particularly interested in the regeneration of neural tissues. So we are mainly working with stem cells that have the capability to differentiate towards cells from the nervous system. So we are working, for example, with neural stem cells or even human dental pulp stem cells. So what we are doing is to combine these cells with scaffolds that have specific mechanical properties, specific degradation rates, and also very specific micro and nano topographies. At the end, what we want to do is to create a support for these cells, so these cells are able to attach, proliferate, and even differentiate towards the cells of the central nervous system. Many times, what we are doing is to functionalize these scaffolds with advanced nanoparticles, such as carbon nanotubes or graphene derivatives, to provide advanced functionalities to our scaffolds, such as electrical conductivity or improved bioactivity. And on the other hand, I'm still working what I started during my postdoctoral stage. So I'm trying to exploit the potential use of polymer capsules in therapeutic applications. And this is particularly the main topic that I will be talking today about the use of polymer capsules as microreactors in therapeutic applications. So with that in mind, I will start with a very brief introduction where I will try to explain what polymer capsules are, how can they be fabricated, and how can they be used as microreactors. And then I will give you a couple of examples where we were using these microreactors in therapeutic applications. These examples will be recent examples where we use this kind of polymer capsules. So, well, there are many ways in which we can fabricate polymer capsules, but in general terms, we can distinguish between two general approaches. On the one hand, those approaches that do use a sacrificial template for the fabrication of the polymer capsule, and on the other hand, we have those approaches that do not use any sacrificial template for the fabrication of the polymer capsule. So, well, in the fabrication of polymer capsules, in the absence of a sacrificial template, the process relies on the self-assembly of amphiphilic block copolymers. Well, as you may know, the hydrophilic block copolymers will have at least one hydrophilic and one hydrophobic block. So when you put this kind of amphiphilic block copolymers in water, they tend to rearrange themselves. They tend to self-assembly so that the hydrophilic block will be oriented towards the aqueous environment, whereas the hydrophobic block will be oriented towards the center of the membrane, a bilayer membrane. Because of their similarity to the natural occurring liposomes, sometimes we coin this kind of structures, synthetic structures, as polymers. And if you think twice, these kind of structures are really interesting for biomedical applications, because we could simultaneously load both hydrophilic and hydrophobic drugs within this kind of polymer vesicle. The hydrophilic drug could be encapsulating within the aqueous cavity, whereas the hydrophobic drug could be loaded in the hydrophobic membrane. And apart from that, and by controlling the hydrophilic to hydrophobic ratio, and also by controlling the self-assembly process, for example, by changing the solvent or the temperature at which we do the self-assembly process, we can obtain other kind of structures other than polymer vesicles that are also really interesting for biomedical applications. And here you have, for example, 3 pictures of different structures that you can obtain by changing all these parameters, such as, for example, round-shaped micelles, worm-shaped micelles, or even more complex structures such as vesicles with gated nanopores. Well, in the case we want to fabricate polymer capsules in the presence of a sacrificial template, here the concept is completely different. In this case, the process relies on the deposition of a single polymer layer or multiple polymer layers on a colloidal template that is subsequently removed, leaving behind a hollow polymer capsule. So a sacrificial template, as you can see in that picture, we can use virtually any kind of colloidal template, either organic or inorganic, with different shapes and also different sizes. So then, once we decide what kind of sacrificial template we want to use, we proceed with the layer by layer, with the deposition of the polymeric layer or polymeric layers. And this usually happens by either electrostatic, hydrogen bonding, or covalent bonding interactions. So once we have deposited the polymer layer or layers on our sacrificial template, we proceed with the template removal. And this will be directly dependent on the kind of template we use, of course. So for example, if we use an a polymeric nano or microparticle as a sacrificial template, such as polystyrene nanoparticles, for example, we could use an organic solvent to remove the sacrificial template. In case we use, for example, silica nanoparticles as a sacrificial template, we could use hydrofluoric acid. Or in case we use calcium carbonate as a sacrificial template, we could use a calcium chelating agent such as EDTA to remove the sacrificial template. So once we have these hollow polymer capsules, we can finally functionalize them by, for example, decorating their surface with targeting motifs such as targeting peptides or antibodies. We could also encapsulate inside active molecules such as drugs or biomolecules or macromolecules, let's say, or we can even incorporate nanoparticles to provide advanced functionalities to our capsules. Well, in any case, these capsules that can be fabricated, as I said, either in the presence of a sacrificial template or in the absence of a sacrificial template, have been traditionally used in the biomedical field as drug, gene, or protein delivery vehicles. Well, in this kind of applications, the polymer capsule reaches the target tissue, and once it is there, it will release the encapsulated cargo. And this release process can be as simple as a diffusion process, or it can be a little bit more complex and be triggered by remote or local stimulus. So imagine, for example, that we incorporate in our polymeric cell light, ultrasound, or magnetic field responsive nanoparticles. Then theoretically, we could remotely activate or stimulate these kind of nanoparticles to promote the rupture of the polymeric shell and allow the diffusion of the encapsulated entity. Or we can make the most of local stimulus, biologically relevant stimulus, such as a change in pH, temperature, solvent composition, or ionic strength to increase the permeability of our polymer capsules and allow in this way the release of the encapsulated drug. But apart from these traditional uses of polymer capsules in biomedical applications, their use as nano or microreactors is gaining more and more interest in the scientific community. And in this case, the concept is completely different. In this case, we don't want the active entity to be released to the media. In fact, what we want to do is to protect this encapsulated enzyme or whatever we decide to encapsulate. We want to protect it from the external environment. And then we want to allow the diffusion and to control the diffusion of reagents and byproducts through the polymeric shell. So the reagent can go through the polymeric shell, react with the encapsulated entity that could be, for example, an enzyme, and then the reaction byproduct will go outside the polymeric capsule. So this is more or less the way the many organelles within our body work. So sometimes we coin this kind of application for polymer capsules as artificial organelles. Of course, here again, we can control the diffusion of the reagents and byproducts through our polymeric cell, for example, by simply changing the thickness of our polymeric cell, or we can use more complex structures, as you can see here in the picture, by incorporating, for example, ion pores or membrane proteins to our polymeric cell that will allow us a selective diffusion of the reagents and byproducts through our polymeric cell. Okay, so after this brief introduction, where I try to explain what polymer capsules are and how can they be employed in biomedical applications, I will start now explaining the first example where we use polymer capsules as microreactors for therapeutic applications. More precisely, I will be talking about the development of antioxidant functionalized polymer capsules to prevent oxidative stress and modulate the extracellular matrix activity in an in vitro inflammation model of nucleus pulposus. This work was published a couple of years ago in Acta Biomaterialia. So, well, reactive oxygen and nitrogen species, such as hydrogen peroxide, hydroxyl radicals, peroxynitrite, and so on, are important cell signaling molecules. And cells are usually able to efficiently regulate their levels by means of several molecules and enzymes with antioxidant capacity, for example, superoxide dismutase, catalase, glutathione, and so on. However, under certain circumstances, such as inflammation, there might be an overproduction and accumulation of reactive oxygen and nitrogen species in the cellular microenvironment that can finally result in oxidative stress. In the particular case of intervertebral disc, this oxidative stress can induce the apoptosis, inhibit the proliferation, induce the senescence, and promote a catabolic phenotype of nucleus pulposus and annulus fibrosus cells, which are the main cells in the intervertebral disc, and finally can aggravate or even promote the intervertebral disc degeneration. So here, with the aim of fabricating or developing let's say, therapeutic microplatform that could efficiently scavenge reactive oxygen species from the cellular microenvironment and protect in this way these nucleus pulposus and annulus fibrosus cells, we decided to fabricate antioxidant polymer capsules. Well, these polymer capsules were fabricated via the layer-by-layer approach, as I will explain before, right now, and they were loaded with catalase. Catalase, as I said, is a potent antioxidant enzyme that is able to scavenge hydrogen peroxide from solution and produce oxygen and water. And apart from catalase, we also functionalize the external surface of our polymer capsules with tannic acid, which is a natural polyphenol which is known to have potent antioxidant capacity. So to test and validate the therapeutic potential of these polymer capsules, we decided to develop an in vitro inflammation model of intervertebral disc. In this model, what we did was to stimulate the nucleus pulposus cells with a pro-inflammatory cytokine interleukin-1 beta. This pro-inflammatory cytokine, after binding to its receptor, it will activate the I-kappa B kinase complex, leading to the phosphorylation of I-kappa B and allowing the translocation of NF-kappa B into the nucleus. As a result of the activation of this mechanism, there will be an overproduction and accumulation of reactive oxygen and nitrogen species in the cellular microenvironment, which will result in oxidative stress, and will also activate or promote the release of several catalytic enzymes such as MMP3 and ADAS5 that could finally degrade the extracellular matrix where nucleus pulposus and annulus fibrosus cells reside. We believe in this project that since we are using or we are developing polymer capsules that specifically will target and scavenge this excess of reactive oxygen and nitrogen species, we will be able to reduce the oxidative stress and attenuate the expression of these major proteolytic enzymes. So to fabricate these capsules, as I said before, we follow the layer-by-layer approach. First of all, we fabricated our sacrificial template. In this case, our sacrificial template were calcium carbonate microparticles loaded with catalase. This is a very simple process. It is called co-precipitation process, in which you simply mix a sodium carbonate and a calcium chloride aqueous solution in the presence of catalase. So the catalase, in this case, this enzyme will be entrapped within these calcium carbonate microparticles. Then after the fabrication of the sacrificial template, we proceed with the layer-by-layer approach by simply incubating alternately these particles with positively and negatively charged polyelectrolytes. And we finally functionalize these polymer capsules with an external layer of tannic acid. Then we remove the sacrificial template with a calcium chelating agent, EDTA, in very mild conditions that allow us to preserve the integrity of the encapsulated enzyme. In parallel, we isolated a primary nucleus pulposus cell from bovine tails, and we stimulated them with the pro-inflammatory cytokine interleukin-1 beta. And finally, we test the therapeutic potential of our capsules in this inflammatory model. So in these scanning electron micrographs, you can see how the calcium carbonate template loaded with catalase look like. As you can see, they are spherical particles, highly porous particles with a diameter between 2 and 3 microns. The initial surface charge of these capsules was negative due to the entrapment of the catalase inside. So then we proceed with the layer-by-layer approach. And as we expected, the zeta potential, the surface charge of our particles was changing from positive to negative. as we were incubating these particles with positively or negatively charged polyelectrolytes. Then we proceed after this layer by layer approach. After coating our sacrificial template with the polymeric layer, we proceed with the removal of the template. As you can see in this scanning electron micrograph, some of the capsules appear collapsed, which is quite characteristic of this kind of hollow capsules. And this happens due to the high vacuum that is used for SEM observation and indicates the successful template removal. This was further confirmed by TM observations, where you can also see that the sacrificial template was removed from our polymer capsules. But to be completely sure that we completely removed our sacrificial template from our polymer capsules, we performed some EDX and FTIR spectra, where we found that after the EDTA treatment, those peaks and those signals corresponding to calcium carbonate were completely removed, suggesting that the sacrificial template was completely removed from our polymer capsules. Well, we also wanted to prove something really important for us. We wanted to prove that the catalase was still inside our polymer capsules after the whole fabrication process and after all the washing steps that this process needs. So for that, we fabricated polymer capsules, but in this case, we used fluorescently labeled catalase. So you can see in these micrographs that catalase was still inside our polymer capsules at the end of the process and at the end of all the washing steps. But more importantly for us was to confirm that this catalase was still active when it is encapsulated or when it is surrounded by this synthetic microenvironment. For that, we first performed some stop flow measurements. In this kind of technique, you can continuously monitor the concentration of hydrogen peroxide in the solution. And we found that those capsules that had catalase inside were able to efficiently reduce the concentration of hydrogen peroxide from the solution, suggesting that catalase was still active when it is encapsulated within our polymer capsules. In order to work with more biologically relevant concentrations, we performed some fluorimetric measurements. And one thing that impressed us a little bit was to find that even those capsules that didn't have catalase inside, but were only functionalized with tannic acid on their surface, were able to slightly reduce the concentration of hydrogen peroxide from the solution, suggesting that tannic acid by itself is able to scavenge hydrogen peroxide from the solution. Of course, the antioxidant activity, the capacity to reduce hydrogen peroxide from the solution was much higher in those capsules that, apart from tannic acid, they also had catalase inside. So from this set of experiments, we could conclude that catalase was still inside, and even more important for us, that it was still active and was able to reduce hydrogen peroxide from the solution at biologically relevant concentrations. Well, we have to consider, however, that hydrogen peroxide is not the only reactive oxygen species that we will find in a real in vivo situation. So here we wanted to prove that our capsules were able to scavenge other important reactive oxygen species, such as hydroxyl radicals. For that, we performed some electron paramagnetic resonance measurements. I will not go into the detail of this specific technique, which is quite complex, but as you can see in this graph on your right, we found that those capsules that were functionalized with tannic acid were able to reduce the levels of hydroxyl radicals from the solution, suggesting that this combinatorial approach by combining the catalase inside and the tannic acid on the surface of our capsules is a valid strategy to scavenge more than one reactive oxygen species that will be really important to provide antioxidant capacity to our capsules. After this initial morphological, physicochemical, and functional characterization of our capsules, we wanted to prove or to test their potential therapeutic application in an in vitro inflammation model of intervertebral disc. So first of all, we wanted to validate our inflammation model. We wanted to prove that what we expected was really happening. As I mentioned before, in our model, we stimulated the nucleus pulposus cells with the pro-inflammatory cytokine interleukin-1 beta. This activates the I-kappa-B kinase complex that results in the phosphorylation of I-kappa-B, allowing the translocation of NF-kappa-B into the nucleus. As a result of this translocation, there will be an overproduction of reactive oxygen species. Some proteolytic enzymes, such as MMP3 and ANAS5, will be also upregulated, And there will be also a higher secretion of several cytokines, such as interleukin-6. So to prove that this model was working as we expected, first we performed some Western blot analysis to check the levels of phosphorylated NF-kappa B. As you can see in this graph, those cells that were stimulated with interleukin-1 beta had much higher levels of phosphorylated NF-kappa B, comparing to those cells that didn't have any stimulation with interleukin-1 beta, suggesting that this model was working as we expected. This was further confirmed when we checked the level of the cytokine interleukin-6. Here again, those cells that were stimulated with interleukin-1 beta had much higher levels of interleukin-6. So this means that our model was working as we expected. After validating our inflammation, intervertebral disc inflammation model, the next thing we wanted to do is to prove that our capsules were cytocompatible, right? So we observed that neither the metabolic activity nor the viability of nucleus pulposus cells was negatively affected by the presence of our capsules. And this indicates that our capsules are cytocompatible and their use is safe for the subsequent in vitro studies. We also wanted to check the interaction between our capsules and the nucleus pulposus cells. And for that, what we did was to fabricate fluorescently labeled capsules that you can see in these pictures in green. As you can see, it seems that some of the capsules are internalized by cells, and this result was further confirmed by flow cytometry, where we determined that more than 70% of the nucleus pulposus cells were able to uptake these polymer capsules after an incubation period of 24 hours. This result was quite interesting for us because we can expect our capsules to scavenge not only the reactive oxygen species that are produced extracellularly, but also to scavenge the reactive oxygen species that are produced inside the cells. So finally, we wanted to prove the antioxidant capacity of our capsules in our in vitro model. We wanted to prove that our capsules were able to reduce oxidative stress in nucleus pulposus cells. For that, we employed the CellRox reagent. CellRox is a cell membrane permeable reagent that becomes fluorescent upon oxidation with the intracellular reactive oxygen species. So we observed that in the case of those capsules that were not stimulated with interleukin-1 beta, the number of CellRox positively stained cells was quite low. However, in those cells that were stimulated with the pro-inflammatory cytokine interleukin-1 beta, the number of positively stained CellRox was much higher, suggesting an interleukin-1 beta induced oxidative stress in nucleus pulposus cells. Interestingly for us, when we treated these these cells with our capsules, we observed that the number of CellRox positively stained cells was much lower, suggesting or clearly indicating that our capsules are able to reduce oxidative stress in our inflammation model of nucleus pulposus. Besides, we also check the expression of major proteolytic enzymes by PCR analysis. In this case, we check the MMP3 and ADAMS5 that are the major proteolytic enzymes in intervertebral disc. And here again, we observe that in the presence of our capsules, the levels of these two enzymes was much lower than in the case of when we were not using the polymer capsules, indicating that our capsules are also able to attenuate the expression of these major proteolytic enzymes that at the end will degrade the extracellular matrix where the nucleus pulposus cells reside. So from this work, we can conclude that polymer capsules loaded with catalyst and functionalized with tannic acid can be successfully fabricated via the layer-by-layer approach by using calcium carbonate sacrificial templates. These polymer capsules, remember, were able to scavenge hydrogen peroxide and hydroxyl radicals thanks to the presence of the encapsulated catalase and the surface functionalization with tannic acid, respectively. And finally, we found in our in vitro inflammation model of intervertebral disc that these polymer capsules were able to reduce oxidative stress and attenuate the expression of two major proteolytic enzymes. Okay, I will explain you now the second and last example where we use polymer capsules working as microreactors for therapeutic applications. You will see that the concept is quite similar to the one that I just mentioned because we again wanted to fabricate antioxidant microreactors. But you will see that the process we followed to achieve these antioxidant microreactors was quite similar. Well, I don't know if you have ever worked with enzymes, but working with enzymes sometimes it can be a little bit tricky. enzymes in the presence of organic solvents, changes of pH or temperature, they can suffer a change in the conformational structure that can finally result in a decreased activity. So in this project, as a synthetic alternative to the natural occurring enzymes, we decided to work with nanoscience. And this work was recently published in Material Science and Engineering C. Well, as I said, nanoscience are synthetic alternatives, right, to the enzymes. They are defined as synthetic nanomaterials with enzyme-like characteristics. In this case, we decided to work with manganese dioxide nanoparticles. These particles are really interesting because they can simultaneously mimic the activity of catalase and superoxide dismutase, that I would say they are the most important antioxidant enzymes that we have in our body. So to incorporate these manganese dioxide nanoparticles in our sacrificial template, we slightly changed the fabrication process of the sacrificial template. Here again, we use the precipitation method by simply mixing calcium chloride and sodium carbonate aqueous solutions. But in this case, we incorporated a little bit of polystyrene sulfonate in this precipitation process. So the resulting calcium carbonate microparticles had a strong negative charge. This strong negative charge was very important for the subsequent incorporation of the positively charged manganese dioxide nanoparticles, which were finally incorporated in our core by electrostatic interactions. So once we fabricated our manganese dioxide decorated calcium carbonate sacrificial template, we proceed with the layer-by-layer approach, as I mentioned before, by simply incubating these particles alternately in positively and negatively charged polyelectrolytes. After coating these coating these particles, we proceed with the removal of the template. We did exactly the same as I mentioned before with EDTA in very mild conditions, finally leading to antioxidant microreactors loaded with manganese dioxide nanoparticles. So first of all, we synthesize and characterize these manganese dioxide nanoscience. This synthetic procedure was performed by Dr. Christos Tapinos and Professor Gianni Ciofani at the Italian Institute of Technology. As you can see in this scanning electron and transmission electron micrographs, these nanoscience were more or less round-shaped and with a diameter around 50 nanometers. And they have a strong positive charge due to, because of the use of polyallylamine hydrochloride during the synthesis procedure. So then we incorporated these positively charged manganese dioxide nanoparticles in our negatively charged calcium carbonate sacrificial template by simply mixing both solutions and allowing the electrostatic interaction to happen. If you look at this scanning electron micrograph, you can see that we didn't find any big difference neither on their size, on their shape of the sacrificial template before or after being incubated with manganese dioxide nanoparticles. And when we check the size distribution that you can see below, we didn't find any big difference between the calcium carbonate particles with or without the manganese dioxide nanoparticles. In all the cases, the particle size was around 3 microns. So the only way we could prove the presence of manganese dioxide nanoparticles on our calcium carbonate template was by XRT. Here, as you can see, the calcium carbonate template, the diffractogram of the calcium carbonate template after being incubated with manganese dioxide present two new peaks at 18 and 38 degrees, which are characteristic of manganese dioxide, and finally confirm the presence of manganese dioxide nanoparticles in our calcium carbonate template. So after the successful fabrication of the calcium carbonate template and the incorporation of manganese dioxide nanoparticles, we proceed again with the layer-by-layer approach. And here again, we found this change in zeta potential of our particles as we were incubating the particles with positively and negatively charged polyelectrolytes. Again, as you can see in the scanning electron micrograms, we obtained the characteristic shape of the hollow polymer capsules. This shape is like collapse due to the high vacuum that is applied in the SEM observation. And again, to confirm the successful elimination or removal of the sacrificial template, we again performed some EDX and FTIR spectra, where we confirmed that the calcium carbonate template was completely removed from our capsules. After this initial physicochemical and morphological characterization of our capsules, we wanted to prove their antioxidant capacity. We wanted to prove that they were still able to reduce hydrogen peroxide from the solution. And we found at biologically relevant concentration of hydrogen peroxide that these capsules were able to scavenge hydrogen peroxide in a concentration-dependent manner. But more importantly for us in this experiment was, or in this project, was to prove the robustness of our polymer capsules. So we repeated this exact experiment, but with polymer capsules that underwent an sterilization process with ethanol. We found that after the sterilization process with ethanol, the antioxidant capacity of the polymer capsules was almost the same, which suggests that these capsules are robust and they can undergo a sterilization process with ethanol prior to any in vitro or in vivo study. And then we proceed with the in vitro characterization of our capsules. And in this case, we use HeLa cells for this in vitro characterization. In this project, we also wanted to study the effect of the surface charge of the polymer capsules in their cytocompatibility and also in their interaction with cells. So for that, we fabricated polymer capsules with either positively or negatively surface charge. And we found quite clear differences between these two kinds of capsules. When we worked with positively charged capsules, we found a clear cytotoxic effect of these capsules when we were working at 100 or 1000 polymer capsules per cell. If you look at this micrograph here in red, you can see strong interaction between the polymer capsules and the cells. If you can see, all the capsules appear like surrounding the cells. And we believe that this is because of the electrostatic interaction between the negatively charged cell membrane and the positively charged polymer capsule. The situation was quite different when we worked with the negatively charged polymer capsules. In this case, we only observed a clear cytotoxic effect when we were working at the highest concentration. And as you can see here in this micrograph in green, the interaction with cells was completely different. It was not as strong as in the case of the positively charged capsules. As you can see, all the capsules appear quite well distributed in all the cell media. Well, when we checked the internalization of our polymer capsules, we found that very few capsules were internalized by cells, irrespective of the positive or negative surface charge. So here we believe that our capsules will mainly scavenge these reactive oxygen species that will be produced in the extracellular environment, and not so much the reactive oxygen species that are produced inside the cells. So finally, we wanted to prove the therapeutic potential of our polymer capsules. And for that, we checked the metabolic activity of HeLa cells in different conditions. In this experiment, we employed as a negative control HeLa cells in normal media and in the absence of polymer capsules. And as a positive control, we employed these same cells, in the absence of polymer capsules. But in this case, we added a little bit of hydrogen peroxide to the media to induce some oxidative stress. So we found a clear difference after 24 hours between the negative and the positive. And this difference was much clearer at 32 hours, which suggests that this hydrogen peroxide in the media is inducing some cell death due to oxidative stress. Interestingly for us, we found that at certain circumstances, such as 10 capsules per cell in the case of positively charged capsules and 10 and 100 capsules per cell in the case of negatively charged capsules, we observe a clear higher metabolic activity of our cells in comparison to our positive control, suggesting that in these conditions, our polymer capsules are able to, let's say, rescue the cells from H2O2-induced death. So from this example, we can conclude that manganese dioxide nanoparticles can be used as a synthetic alternative to natural occurring enzymes, such as catalysts. These polymer capsules are able to efficiently scavenge hydrogen peroxide at biologically relevant concentrations. And we also found that the surface charge of the polymer capsules will determine the interaction with cells. Remember that positively charged capsules had strong interaction with the cells, but they also promote a more cytotoxic effect. Finally, we found in an in vitro H2O2-induced oxidative stress model some therapeutic effect of our polymer capsules. So, well, now we'll talk a little bit about our future work. Now what we are trying to do is to fabricate multifunctional polymer capsules. If you think about the examples that I just mentioned, we are targeting one single mechanism, one single mediator in our pathology, right? In this case, it was oxidative stress. But targeting one single mechanism, mediator, or molecule ignores the complexity of the complex disease microenvironment. So in order to target more than one mechanism, we are working now with multifunctional polymer capsules. So these multifunctional polymer capsules will be loaded again with an active entity that could be an enzyme, but in this case, the surrounding polymeric shell will not only provide structural integrity, it will also allow us to release a drug in a pH, or in this case, in a biologically relevant stimulus. And this work we are doing in collaboration with Professor Marcelo Calderon and also with the help of Dr. Neha Tiwari from Polymath. So we are achieving this by using dendritic polyglycerol, functionalized dendritic polyglycerols that we are using as a polymeric layer. In this way, we believe we will be able to target more than one complex, more than one molecule or mediator in the pathophysiological microenvironment, and we believe that this could be better than the current treatments that sometimes they fail whenever we are trying to treat a complex pathology. Well, finally, I would like to acknowledge all these people that actively participated in all the projects that I mentioned. In particular, I would like to acknowledge Edurne Marin, who is doing the PhD with us, and well, she performed most of the experiments that I presented in the second example. Of course, I would also like to acknowledge the funding agencies for providing support to these projects. And of course, thank you very much for your attention. I have to say again that this was a very nice opportunity for me to discuss a little bit about my work. So thank you very much for this opportunity. And I will be very happy to answer any question that you might have.
Thank you, Dr. Eiter, for your nice presentation. Very impressive. And you presented multiple polymeric reactors using quite an interesting chemistry, inorganic chemistry, using calcium chloride and sodium carbonate, and encapsulating multiple small molecules, which are actually important for reducing the oxidative stress or as a therapeutic effects. And I'm sure that you are working on to something very interesting application from here in biology. Although you showed various effects using interleukins, and you already have gone into the deeper biology of an inflammation. So before few others ask question, it is pleasure to see your students and Professor Marcelo Calderon's team here today attending this talk, including School of Pharmacy, MIT, Pune. We have many PhD students and professors from various other institutes and university who have actually been participants. We touched participants today that states actually how interesting and popular this talk was in our community. So I have two questions, small questions, that which polymer templates you could use or you have used actually other than what you have been using, and what is a maximum size that you could reach from, say, 50 nanometers? Can you reach to few micron size? Because I might find a very interesting applications. What is the stability of these reactors in acid as well as base pH, considering as a GIT tract pH in stomach as intestine? So let me ask these two small questions first, that which other polymer templates can be used for the purpose that you have been using for encapsulating small or large molecules as a cell-passable structure? And the second is, what is a greater size and stability of these reactors?
Okay, thank you very much for your kind questions, Professor Kandari. Regarding the polymers that you can use for fabricating these polymer capsules, I think you are talking about the shell, right?
Yeah.
Well, you can find in bibliography many kind of examples. Most of the times they are using synthetic polyelectrolytes, as the one that I presented, such as polystyrene sulfonate, polyethylamine hydrochloride, and these kind of synthetic polymers. But in the project that we are sharing with Professor Calderon, we are trying to use also polypeptides due to their biodegradability. So almost any kind of thing that it can be deposited on the top of your template by electrostatic, covalent, or hydrogen bonded interaction can be used. So the possibilities are many. Then about the second question about the stability and the size of the capsules that you can achieve, this is a question that they always ask me because, well, it is true that if the interaction is electrostatic, a change in pH will result in the disassembly of this polymeric shell, right? And this is true, but at the pH that we are working in our biological applications, we never found a problem of stability. And in case you want to improve the stability of these polymeric capsules, you can always go for a cross-linking reaction between the polymeric layers. For example, between the amines and carboxylic groups in the polypeptides, for example, you can easily perform a cross-linking reaction to provide more stability to your capsules. And in terms of the size, well, the largest particle that I have ever made was 3 microns, 3-4 microns with calcium carbonate microparticles, but I don't see any problem to do slightly bigger particles by using other kind of templates. Of course, in this case, you could go for polystyrene microparticles that are commercially available. So I usually see the problem when you try to go to smaller particles.
Thank you. And I think before somebody asks a question, I have my last question. Is typically you have shown small molecules like tannic acid and some other biologically relevant molecules in for the inflammation. Have you tried actually multiple molecules encapsulation, say small or the larger or the combinations, and how do you find their release systems or the functionalities that is actually changed?
Yeah, that's a really nice question, and this is precisely what we are trying to do now. Because of course, if you incorporate a drug or you change the structure of the polyelectrolyte that you are using for the fabrication of the polymer capsule, then that could have an impact on the stability, and that is something that we are trying to study now. In the first example, it is true that we combine a large molecule such as catalyst that is 250 kilodaltons with a small molecule tannic acid. So this is more or less the procedure that we want to follow to go for combinatorial approaches that can target more than one mechanism, because targeting one single mechanism is quite a simplistic view of a complex pathology.
I'll request any other questions from the participants. Marcelo, if you want to ask question, because you are staying far away.
Hey, it's fine. There are already 2 questions on the chat box. There is one question from Patricia Brackbat. Sorry, for sure I didn't pronounce it right. He's asking if wouldn't PAH be toxic?
Which one? Toxic?
PAH.
I never found a cytotoxic effect of PAH. Maybe if it is released, it can induce some toxicity. It is true that if PAH at the latest surf layer, and as I saw in that slide, you found some cytotoxic effects. So it is true that the strongly positively charged polyelectrolytes are known to be cytotoxic. So this is something that you have to always consider. That is true.
Yeah, and then you have Marcelo, your question, please.
Yeah, I was just wondering if you can have any strategy where you can enhance the interplay between the antioxidative activity from tannic acid and catalase.
I never checked that, to be honest. I think what we try to do is to maximize the amount of catalyst that we encapsulated, and we found that when we try to encapsulate more, we completely lost the possibility to create the template. So I try to maximize, but I never try to check this interplay between the surface functionalization with tannic acid and encapsulated catalyst. But this is something that we could study in the future.
So how large the molecules could be encapsulated? What would be the, say for example, have we tried to look for the protein affinity and serum albumin? Very simple protein, stable protein.
Well, here usually the general trend is to think that the bigger the particle, the better it will work. Because if you try to encapsulate molecules that are too small, they might be loose by diffusion through the polymeric shell. So catalyst is quite a big molecule, as I said before, 250 kilodaltons. So here we were safe, and we knew that it was not going to be leached out from our capsules. So I don't think there is any problem to try to encapsulate bigger particles. I have seen in bibliography people encapsulating antibodies, bovine serum albumin, which are also quite large molecules.
And maybe this is also Marcelo's question that can you have a stimuli sensitive polymeric capsule reactors using that are very well known?
Sorry, can you repeat the question, because there was a problem with the connection?
Right. So, say for example, this is a very well based SAID platform that you have worked. I'm trying to say that the release of these entities will be dependent on various buffers, right? But
yeah.
can you have you started working on preparing those capsules which will be stimuli sensitive? For example, a specific pH they will actually be very stable, and at some other pH they will not.
No, I have never done that, because my main aim is not to release the encapsulated drug. My main is to keep it inside. But of course, by changing the interaction between the polyelectrolytes, or by using cross-linkers that are responsive to pH or even more biologically relevant stimulus, such as metalloproteinases, you could control the release of your drug, of the encapsulated entity.
So there are many things that actually you can do. Temperature sensitive, maybe I should not inform you, but you already have those multiple thought processes. Looks very interesting, very fascinating. There's a next question from one of a participant. For designing, say, to retentive drug delivery system, what chemistry or polymeric would be used? Suppose you want to use these capsules for the oral delivery. Can you do that, and what changes you might actually suggest?
I'm not an expert in this kind of application, to be honest, but I assume that in the gastrointestinal environment, correct me if I'm wrong, but the pH has to be low, right? Will be low.
Very highly acidic, yes.
Very highly acidic. So I think that any polyelectrolyte, any kind of interaction between layers that relies on the electrostatic interaction will be broken at very acidic pH. So I think that almost anything could work. And of course, if you are thinking about a biomedical application, when you are thinking about the release of a drug, I think that something like a polypeptide could perfectly work because of their biocompatibility and biodegradability. So, for example, in the case of Marcelo, we are working with 2 polypeptides, and because of that reason, right? Because they are biodegradable and biocompatible.
Right, and you could also use a targeting of your encapsulated entities actually when you protect and when you actually allow them to diffuse and then permeate, and then probably you can send multiple things. I think that's a so the control over the size from nanometers to micron is a very flexibility over there. There's another question, Dr. Eiter, which says that are these systems very identical to nanocochleates that are used, or they are known actually? They are in the rod shape, and what you suggested are the spherical ones. So are you aware of this difference?
Well, as I mentioned, in the layer by layer approach, you can use virtually any kind of sacrificial template. So you can change the shape of your capsules. And there are several studies where they found that rod-shaped particles or capsules are better internalized by cells. So this is something that you can also play with it, right? If you change the shape, not only the size, but also the shape of your capsules, maybe you can promote a better internalization of your capsules, if this is the thing you want to achieve.
So what would be the mode of delivery? So will this be a systemically delivered? How do you actually use them in your say in cell studies? We understand you might spike these capsules, right? But say suppose if you move on into animal studies, which you might have done, would you like to share those content?
Yeah, the systemic delivery is something that right now I don't see it feasible, right? Because I assume that in the bloodstream they will suffer a huge protein absorption, fouling of proteins, and at the end you will destabilize the colloidal suspension of the particles. We are thinking more in the local delivery. In the project that we are sharing with Professor Calderon, we are trying, or what we will try to do in our in vivo studies is to inject these particles directly in our target site via a catheter. We want to place these capsules in the heart. But the
Very
systemic
good.
delivery, I find it quite difficult because of, as I said, the falling of the proteins and also the immune system that will get rid of them or might get rid of them.
It's very exciting. Will you allow us to actually make a stability if we work together in a say physiological pH, and as we work with Marcelo quite often, I think there is a great possibility to make them stable in a not only GI pH but maybe systemically driven, because this becomes an that this could be an alternative that is achieved over to liposomes and nanoparticles if they show stability in a systemically delivery system.
Exactly. Yeah, that will be great. And as you said, this could be an alternative to this system that they also have their drawbacks, right? Like liposomes with their stability or their difficulty to encapsulate some macromolecules.
So have you covered this as an IP patent or something so that...
No. I mean, the academia, and we are not so focused, at least in my group, we are not so focused in the patents. We are more focused in publishing research papers.
So you have kept that space open for us to breach in. I hope you file your patents in that. But I think the context, the philosophy of bringing you here is to understand, the difference of our thought processes. And this really is very exciting to, know how differently the researchers like you and Marcelo is thinking over to what we think. So that is very interesting aspect of it. I would open other questions, please. Ashwin, you could actually unmute mic so that if anyone is keen, they might. We have here Dr. Preeti, who works very well in the enzymes area. She might have a question.
Yes, I'll do it.
We are party. If Neha is there, she can ask question. If Neha, you are there. We see some of our startup companies members here. Dr. Allen, who works in an immunology area. We have here Balram and Smriti actually, who are working with Marcelo. So may I request your questions, please?
I can ask while the people is still thinking their questions.
Yes, please go ahead, please.
I have a question about the second project. I thought this is quite fresh. I didn't know it. I mean, you just published that. I noticed that you have a different surface charge on the particles, whether they have the manganese oxide inside or not. Is this an indication that the manganese oxide is sitting on the surface, or what do you think about that?
Let me go back to that slide.
Yeah, I think it's number 28.
I don't know why I cannot.
So I told you, Marcelo, you both are living very different distance, long distance. So this platform helps.
Yeah, true.
Right. Can you still see my presentation?
Yes.
So yeah, let's. Oh.
This one, yeah.
So here, what we found that if you see the template, initially it had negative surface charge because we use polystyrene sulfonate for the fabrication, and as we incubated with the manganese dioxide nanoparticles, it changed to +5 because of the positively charged nanoparticles within our template, right? But then if you look layer after layer, I wouldn't say there is a huge difference between the zeta potential, right?
No, but they changed the sign, right?
Yeah. This is something that I, to be honest, didn't realize. It is something that I have to check better because I was not aware of that. But yeah, there might be a slight change that can be because of the presence of manganese dioxide nanoparticles that are somehow interacting. I have to say also that checking the zeta potential of these layer by layer capsules became quite tricky as you increase the number of layers. So there is always an interaction between the previously deposited layer and the layer on the surface. So if you really want to check that, you have to be very careful with the conditions at which you perform the experiment.
But Marcelo was asking, maybe that's what I understood, that where would be your magnesium, Mn that will actually be, will it be on the surface with the calcium and your template of sodium carbonate, or?
Well, the calcium carbonate template is removed, right? So
Okay.
I imagine for that I could tag the manganese dioxide with some fluorescently labeled molecule or try to do some TEM that we didn't do in this project. But I believe it will be within the polymer capsule, but attached to the inner cavity because of the electrostatic interaction. You have to think that manganese dioxide is positively charged and the next layer will be negatively charged. So there might be some kind of interaction, but this is something that we could check by TEM, for example. I don't know exactly where they will be. I assume they will be inside the polymer capsule, but I don't know exactly where, in the middle or just a little bit closer to the polymeric shell. This is something that we could check by TEM.
You showed the paramagnetic trait of the, understood what position of checking the magnetic trait of these materials. In one of a slide, I don't recollect, 14, 13 maybe. But
the electropharamagnetic resonance. Okay.
Right. So EMR was shown there. Yes, I think. Yes. So that magnetic field, which is on your x-axis, which is very indicative of high magnetic field. So where does this magnetic field is coming from? Is it coming from?
No, in this experiment is quite similar to NMR analysis, but you are checking the other aspects of your in this case, the hydroxyl radical, right? This experiment we only performed to confirm that our capsules were able to scavenge hydroxyl radicals. So the hydroxyl radicals, when you bind them to a paramagnetic molecule, they give you this kind of signal. And then you check how this signal is being reduced as you incubate with your capsules. And this means that the hydroxyl radical is being scavenged. So it's not talking about the paramagnetic properties of your capsule. It's talking about the paramagnetic properties of the hydroxyl radical binded to this molecule. That, to be honest, I don't remember which one it was because I did it few or 3 years ago while I was in Switzerland. I don't remember the name, but this is how it works. It's quite a complex technique. It's similar to NMR, but you are checking the radicals in this case.
Right. So I think I got a little bit confused with the paramagnetic property. I think this is a scavenging of an hydroxyl
exactly.
mole, right? That's exactly what you are trying to do.
Exactly.
So let me see if there are new questions now that have come. One question. Have you tried to actually feed these nanoparticles to bacteria or using antibacterials? So there is a lot of space remaining to explore these entities.
Exactly. Yeah, no, I have never worked with bacteria before, but I think that in the group of Professor Calderon, they are trying to do some antibacterial application for these kind of capsules. But in my case, I never worked with bacteria.
So that was a question coming from Professor Sangve, who works in this close area. Maybe I will connect you both after this presentation.
Mm-hmm.
So, audience, any other questions? Ashwin, you have a question? Yuraj? So,
hi, this is Alain D'Souza. Dr. Larania, good afternoon. Hi. I just had a question on more on the immunology aspect. When you all did your experiments with interleukin 6, did you all use any other pro-inflammatory cytokines or mediators as a readout? And did you all only do your experiments in vitro, or was there an in vivo model with confirmation as well? Thanks.
Okay, thank you very much for your question. So far, I've only worked in vitro models, right? I come from a laboratory that is mainly focused on materials, and I had my first step in this biology field a few years ago, let's say. So I started working with in vitro, and I hope with the next project we will be able to go to the in vivo situations that will tell us much more information about our capsules. And in terms of the interleukin-6 that you mentioned, this was just to prove that our model was working as we expected. So we, on the one hand, checked the phosphorylation of NF-kappa B, and on the other hand, to complement this experiment, we also checked the secretion of interleukin-6 that we know it is overexpressed by the NF-kappa B pathway.
Great.
Thanks so much.
You're welcome.
So, Dr. Eiter, if you want to say more comments on these questions, we can take your own comments in addition before we close.
Okay, no, I think that they were really interesting comments, and they make me think a little bit. It's, as we said, food for thought, right? Now I will be thinking about many of your questions, and I hope I can answer many of them by performing experiments. But of course, doing an in vivo will be vital to see the real potential of these capsules, to see how they interact with the immune system, as the last question asked. And also to check how they interact with in the bloodstream, for example, how the biodistribution is. All these kind of questions, I think they will be answered whenever we do an in vivo study that we hope to do it quite soon, if we can. And again, thank you very much for this opportunity. I think all the questions were really interesting, and as I said, they helped me to think more about my system and how can I improve it.
That was not the objective, but we have a very interesting topic, which was very exciting. And I must thank Marcelo for introducing you to, present this webinar to me and my colleagues, both from academia as well as startup companies. We find very exciting participants actually arriving from many places. They come from national labs, and they are from the private institutes. They are from various places. So this was very rare of the response that we received in this lockdown, which crossed more than 100. So thank you, Dr. Eiter, for your time. And thank you for your nice presentation. This doesn't close our interactions through the webinar. In fact, this opens, and Professor Calderon is a great interface over there. And hope to see you soon in your lab when all things are actually open. I was going to see you and Marcelo in October, but that doesn't look possible in current scenario. So on behalf of School of Pharmacy, Maharashtra Institute of Technology, World Peace University, finally, I thank you very much for your presentation today, and take care.
Okay, thank you very much.
Thank you all the participants. We can close the meeting now.
Thank you very much. Bye-bye.
Thank you.
Bye.